project actually started a couple years ago. I was doing an acoustic telemetry project down in New York Harbor, and we kept getting these really annoying Atlantic sturgeon detections that really just did not match with the time of year that we predicted Atlantic sturgeon to be down in New York Harbor. So it took a little bit of time, but I was talking with Amanda, and we just kind of kept getting this hunch about something was more to this story than just fish doing weird things. So we have been dusting off the data, kind of looking at it from a little bit of a different perspective. And when we did that, we started to get some new insights into Atlantic sturgeon movement behavior, but also starting to look at some new vulnerabilities that the population might be exposed to. So a little bit of background, Atlantic sturgeon, there's 18 genetically distinct populations of Atlantic sturgeon distributed across the east coast of the United States and Canada. Most of those populations are currently listed as endangered, and a lot of their conservation challenges has to do with some of their unique life history attributes. The one that matters for today's talk is the fact that this is an anadromous and highly mobile fish. The adult Atlantic sturgeon, when they are born, when the babies they're born in freshwater rivers, they migrate out to the open ocean. And once they get out there, they tend to make these very, very large coastwide migrations. The figure on the right there, that's showing you just the detections of Hudson River Atlantic sturgeon. And you see that those are fish going from Maine all the way down to Florida. This is a truly coastwide distributed fish. Now, Atlantic sturgeon, they do spend the vast majority of their 60-plus year life cycle out in the ocean, but when they are ready to spawn, they need to go back to some sort of freshwater river. Generally speaking, they go to their natal river in order to spawn. They spend a couple weeks there, and then they head back out to the ocean, repeat that process all over again. Now, these spawning migrations, they do tend to be really predictable. So in the Hudson, we look for Atlantic sturgeon to arrive somewhere around mid-May. They stay again a couple weeks, maybe a month or two, and then head back out to the ocean. So again, this really highly migratory strategy, it makes conservation extremely challenging because it is very hard to conserve and manage a moving target. So when fish are out in the ocean, we don't really know where they are most of the time, and so it's really hard to really enact good conservation and management if we can't figure out where and when fish might be running into problems. But when fish are in the river, when they're on those spawning migrations, there is that very small and critical window where fish are in these rivers. Those are areas where we can better quantify and understand their movement, and we can understand and control human behavior a lot better in these river systems as well. So a lot of what we do for adult conservation is try to protect that spawning population. So we do things like time of year restrictions or other permit conditions to really limit how much humans can influence Atlantic sturgeon when they're on those spawning runs. This is all well and good. It tends to be really effective, but it does have one really critical assumption, which is that we know when and where Atlantic sturgeon are spawning. And that turns out to be a little challenging because a lot of what we know about Atlantic sturgeon movement is based on historical observations. These observations might not be up to date with environmental change, and they don't necessarily account for more modern technologies, things like acoustic telemetry. So as we start to see some of this variation, right, like I saw those fish down in New York Harbor, we also see other things that don't quite make sense with what we at least formerly believed. We start to wonder, is this variation we see actually important for conservation, or is it just kind of random noise in the data? So fortunately, we're able to address this because we have access to what is really a very unique and unprecedented resource in the New York State DEC acoustic telemetry array. This array, it's about 80 ANOVA-C receivers that are distributed from Albany at the Troy Dam up north all the way down to New York Harbor, and as time has gone on, we've continued to expand to some nearby waterways like Newark Bay and then out towards Long Island Sound through the East River. These receivers at this point, they are maintained year-round and have been out for over a decade at this point. So we have a detection database of over 32 million acoustic detections, really just underscoring that this is a massive database we can use for understanding fish movement and behavior. So looking at this map, you'll see maybe that there's two areas that I've kind of color-coded to highlight. This first area in blue, this is Hyde Park. These areas are going to be important throughout my talk. Hyde Park, it is the only known spawning location for Atlantic sturgeon in the Hudson. And so when we talk about Atlantic sturgeon spawning, we think that fish go to Hyde Park, they spend a little bit of time there, and then they go back out to the ocean. This other area, highlighted in yellow, it's Catskill. Catskill, there is historic reports of there being an adult fishery there. It did, at least formally, we think, support spawning, but today we're not exactly sure the ecological relevance of this area. But just generally speaking, when we try to do any sort of movement study in the Hudson, we do try to keep at least an eye out in Catskill to try to figure out what's going on there. Now that acoustic detection database, it is truly one of a kind, but it really is only as good as an accompanying tagging database because we need to have some way to kind of reference a detection with a species and then some biometric data about that tag. Now, because we do work with a lot of anadromous fishes, fish that could be tagged anywhere throughout the entire coast, we do depend on researchers throughout the entire eastern seaboard to kind of contribute their tag codes to us in order to build a tag database. So over the last couple years, I've done just that. Working with folks, we have a tag database of about 3,400 individuals. Most of those are Atlantic sturgeon. Generally speaking, they're adult Atlantic sturgeon. And we also have genetic data on a good number of those. I'm not going to go into the details, but with the genetic data, we can tell if a fish was born in the Hudson or in one of those other populations I mentioned. And we can also determine its individual sex. So this gives us a lot of that biometric data so that we can refine any analyses by things like sex, natal origin, or age class. So when I took that big detection database, I filtered it down to Atlantic sturgeon. I looked for fish that migrated upriver of New York Harbor to get into the Hudson River, and then I'm looking at data from 2016 to present. So still a pretty robust, almost decade-long data set there. So as we start to look at individual differences in movement, we have kind of three basic fundamental questions. First, is there variation? Then we want to know, is that variation structured by space or time? And then finally, is variation or behavior repeatable across years? And this one's interesting because if there is that variation and we see individuals kind of repeatedly behaving differently year after year, we start to think it's not just kind of environmental variation or noise, we start to think there might be fixed phenotypic differences in the population. And then once we answer all those questions, we can actually get at the final question, which is, does this variation actually matter for conservation? So just kind of stepping through the analyses I did to kind of address these questions. The first thing I needed to do was just describe behavior in a way that would capture meaningful differences among individuals. I eventually settled on these four metrics here. They kind of represent both a space and time continuum to look at some of these differences. And they included first and last date of detection in the Hudson River, the number of days a fish was in the river, and then the maximum upstream migration distance. I know these metrics, they're kind of coarse, but my thought was that if we do have very distinct differences, these metrics should be enough to capture those and separate fish, while also being very, very relevant for conservation. Once I defined those metrics for each fish and each year, I used a Gaussian finite mixture model to actually group fish. Again, just trying to statistically group fish that behave more similarly to one another than they do the rest of the population. I then tried to see if group membership correlated to some of that biometric data I talked about earlier, size class, sex, and natal origin. And then finally, that repeatability piece, for reasons I'm not going to get into, that is actually like a really complicated equation that you need to use to get at repeatability. It's there, I can tell you about it later. But what's only important for right now is that if you look in the literature, a value of R, which is repeatability, if it's over 0.5, it generally indicates extremely strong repeatability in a wild population. So looking at the results, I created this figure here where we have date on the x-axis and the river kilometer on y. And then I have sort of on the side here just some areas of the river so that if you're familiar with the Hudson, you can kind of place the river kilometer to an area in the river that I'm talking about. When I looked at the results, what was very, very clear was that we had very strong support for two very distinct phenotypes. The first phenotype is what I call the short residency phenotype, and these fish behave exactly like we predict Atlantic sturgeon to behave in the Hudson River. These are those fish that you can tell from this plot. Again, the color here, it's going to kind of indicate an increase of detections. But these fish, they arrive kind of on average in mid-May. They come to here. This area of yellow indicates kind of the Hyde Park spawning area where they congregate for a couple weeks. And then they leave on average around mid-June. And what's noticeable is that when fish leave Hyde Park, they pretty much immediately leave the river. They don't hang around in the river any longer. They don't go, on average, up or downstream very long. Once they're done, they head back out to the ocean. And what's also interesting is we see this kind of secondary peak here of detections, which is in Catskill. Looking across the data across all years, we do see repeatedly the same fish going to Catskill every year. There's not a ton of fish in my sample that do that. I think we've kind of under-tagged the fish that have that behavior. But it is noticeable that they, for now, at least group with that short residency phenotype because of the timing of when they arrive and depart from the river. So again, this is the quintessential Atlantic sturgeon spawning run. But we had one more phenotype, which I'm calling the extended residency phenotype. These fish, they tend to get to the river about a month after the short residency fish. And then once they're there, they have a much more protracted and a much longer duration of residency in the Hudson. What's noticeable, I think, is that if we look at Hyde Park, again, that spawning reach, there's really limited overlap between the two groups. As fish in the extended residency are arriving to Hyde Park, fish in the short residency are actually leaving. So they're not really overlapping very long at that, at least reported or supposed spawning area. And then what's also noticeable is in the extended residency group is that once they leave Hyde Park, they don't actually leave the river. Most of them kind of congregate around river kilometer 130, which is Diamond Reef, for those who might be familiar with the Hudson River. A diamond reef was really not on our radar as being important Atlantic sturgeon's habitat, and it actually seems to be one of the most important habitats in terms of fish being there for the longest duration of time relative to any other habitat in the river. And then, like I keep saying, once fish got to Diamond Reef, they then stayed like 3 months before finally leaving sometime around mid-September. So a much longer duration of occupancy than we would have expected. And then finally, that repeatability piece. Repeatability was extremely strong, 0.52. And to put this kind of into perspective, I did look to see how often a fish would switch between each phenotype, and it was really rare. So 97% of the time, if a fish assigned to one phenotype, it stayed in that phenotype for up to 6 to 8 years of detection. So it doesn't seem to be fish that randomly switch back and forth. It seems to be something that's very fixed and stable over time. Looking at some of those correlations to the biometric data, there was nothing meaningful here. So size, sex, and natal origin did not consistently predict cluster membership. So it doesn't seem to be just an easy explanation for why fish might belong to one phenotype or the other. So going back to that overarching research objective, does this variation matter for conservation? That answer is obviously a little bit in the eye of the beholder, but I'm certainly going to argue it does. I wouldn't be here if I didn't think so. But I think that kind of thinking about this from a more ecological perspective, we know that functional and phenotypic and genetic diversity, those are all things that are the backbone of resiliency. And I think it's important to note that the Hudson River, it is the largest population of Atlantic sturgeon on the East Coast. And so it's very possible that some of these forms of diversity we're seeing are really contributing to the recovery success of this population. It's really hard to know how this resiliency might be coming into play. For example, because these two phenotypes don't directly overlap at the spawning ground, it could be that they have a longer duration of spawning, reducing the risk of complete year class failure. That is something we see down south all the time, but it might not be as prevalent or as common here in the Hudson. Because these two groups are kind of spatially segregated for most of the year, they could also be kind of occupying slightly different ecological niches, reducing competition, and allowing each group to kind of exploit resources slightly differently. And then also, it could be that resiliency is something that we're benefiting from now, but it was really something as a byproduct of a while ago, back when the fishery was open. That fishery, it really targeted fish at Hyde Park. Probably, my guess, is the short duration phenotype. And so because we have multiple phenotypes in the group, it could be, again, we just weren't able to overfish any single phenotype, allowing again more recovery success now. So again, we don't know why potentially this resiliency is occurring, but it is something that we have to think is at least helping the population out. Now the flip side is that with this diversity comes those expanded threat windows. From a spatial perspective, that short duration phenotype, we now are increasingly looking to Catskill as being an area of occupancy. It has to have some sort of ecological significance. We do think they are probably spawning there. We're going to look into that this year, hopefully, with some other technologies to see if we can capture spawning going on. And then if we look at that extended duration phenotype, we have an extended use of Hyde Park, but then now also occupancy of Diamond Reef. We have now thinking about space, we have more space occupancy, and then this kind of plays out across an 88-day time window between when the short and extended duration fish leave. And 88 days doesn't really seem like a ton of time, but in a system as dynamic as the Hudson, I think 88 days could really change the vulnerabilities of any one population. And then finally, the last thing is a bit of a dramatic, but I think important message. These individual fish, these phenotypes, they've been swimming around for decades. And we really did not kind of hear the story that they were trying to tell us. But as we use more modern technologies, things like acoustic telemetry, we're able to paint a much, much clearer picture as to what these fish are doing and the vulnerabilities that they might be exposed to. And once we see that picture, once we kind of understand the story a little bit better, it's a little irresponsible to kind of turn our heads and ignore the data that we're getting. I know that that is a tall order, right? Fish management, it's not easy by any means. But we, as fisheries biologists, we really manage for the average individual, and that's worked pretty good till now. But as we continue to see a lot more variability in our data, the ideology of managing for the average, I think, is going to become increasingly less successful. And we're going to continue to fall short of fully protecting all the diversity that exists in these populations. So I do think we are kind of at a little bit of a crossroads right now where we can protect these fisheries using our new technologies and preparing these individuals for a new climate. Or I think we're at a real potential to be watching some of these populations slip away to history. With that fun message, I will be happy to take any questions.